Yeah, everyone's in now, Roger.
All right, good evening, fellow beekeepers. Welcome to this BIBBA webinar. The speaker this evening is Jacques van Alphen, and welcome to you, Jack.
Thank you.
Thanks very much for agreeing. Yeah, thanks very much for agreeing to. You've unmuted. No, you. What are you doing?
We can hear you, Roger.
It's okay, I think. Go on.
Okay. Yeah, Jacques is a retired researcher of many creatures, not just honeybees. He is actually Dutch, but he keeps black bees in the north of France, which no doubt he'll tell you just a little bit about. I've been keeping bees now for 60 years, and the honeybee losses are far greater than when I started. I've always thought the reasons were more than stated. Then I saw a recent paper by Jacques called Why Honeybees Are Dying, A Forgotten Cause. This presentation should give answers and a possible cause that's probably a bit different than the standard ones we're usually fed. Now, Jacques will be speaking at a National Honey Show in 2024, but this evening he's speaking to us about the bees that are dying. So can I hand over to you, please, Jacques?
With pleasure. I'll look for my... Okay. I
But
think...
better point out, sorry, but better point out first that if you've got any questions, please use the facility at the bottom of the screen. Thank you.
Okay, good evening, everybody. I've slightly changed the title of my talk, but it's the same talk nevertheless. I thought it was a little bit more clear what happens. The story I'm going to tell is a rather long argument, and you need all the different parts of it. So please, when you lose me, give me an alarm so I can make sure that you get everything that you need on the way to understand the whole talk. The problem started for me when I realized that we have more than 40 years after the Varroa invaded Europe, and most honeybees are still not resistant. And then I was looking in the literature, and I discovered that both in South Africa and in South America, natural selection was very quick in making the honeybees resistant against Varroa and the associated viruses. And so why is this difference? And I was thinking about this, and then first I thought it was a trivial problem, because of course beekeepers do everything possible to prevent natural selection through their actions. But after thinking a little bit more, I realized that that is not the whole story. It certainly is important, but you cannot explain everything by that. Now, before I continue, I'm a biologist, and I'm using terms from evolutionary ecology and from genetics. And I thought it might be wise to tell you first what all these words are meaning. We will be talking about panmictic populations, and a panmictic population is one in which every individual has an equal chance of mating with every other individual. And maybe you shouldn't take that literary, but at least there should be an enormous mixture of genetics in such a population. And then we'll talk about a polyandrous mating system. And of course, you all know that honeybee queens mate with many drones, and that's called polyandrous. And then at some point, we'll very shortly mention eusocial, and people always think, well, is social not enough? So I've defined that as well. Living together in societies in which animals work together to raise offspring with overlapping generations and divisions of labor. So the honeybee is eusocial. Now some terms from genetics. We will talk a lot about alleles, and alleles are nothing more than variants of genes. And they are recessive when you need 2 copies to make them visible. You need 2 copies to get expression of that allele. But the dominant allele is always expressed. And so that's when you have only 1 copy, you still see it expressed. I will explain it a little bit further on. Then an individual that has 2 copies of the same allele is called homozygous, and an individual that has 2 different alleles of a particular gene is called heterozygous. Very important today is the word recombination. It is the creation of new genotypes through the exchange of pieces of paternal and maternal chromosomes. Now, those are the terms. To do the genetics, we can see that in one slide, and it's handy to think about people with blue eyes and with brown eyes. Blue is recessive. It means you need 2 copies to have blue eyes. And brown is dominant, so you can have brown eyes if you have only 1 copy of that, only 1 allele for brown eyes, and the other could be blue. So that is one of the reasons why parents that have brown eyes can sometimes have a child which has blue eyes. And then if you can at the same time see that the blue-eyed individual is homozygous, and the brown-eyed individual could be either heterozygous or homozygous. Now recombination. The queen in each cell has 2 sets of chromosomes. One comes from the father, from the drone. That was her father, and the other comes from her mother. And when she is going to make eggs, then she has to make cells that have only 1 copy of the chromosomes. And then these chromosomes, they come together, they pair, and then they might exchange pieces of the chromosome, which means that in the eggs, there could be genetic combinations that did not occur in the mother. So you make new genotypes. Now we're going on with our problem. It's interesting to note that in all the cases, published cases where natural selection resulted in resistance against Varroa, you find that there are 3 important characteristics. One is that these resistant bees, they have Varroa-specific hygienic behavior, which means that they can smell whether a cell with a larva or a pupa of a worker contains reproducing Varroa. And if they smell that, then they react by opening the cell and removing the larva from those cells. And then the second character that you find is that they are very effective in grooming both themselves, but also others. And by that, they can remove Varroa mites from adult workers. And then finally, because generally bee colonies die not directly from Varroa, but generally because of the virus infection that has been brought by the Varroa mite, resistance to viruses is also found in these populations that have become resistant by natural selection. Now, let's look at things that we cannot easily explain with the idea that beekeepers avoid natural selection to work because of they do all sorts of things. And one of these, the most important problem I saw comes from an experiment that was done in Sweden on the island of Gotland on the southern tip. There is a peninsula which is rather isolated, and Ingmar Fries did a large experiment, and it's the only experiment which is really very nicely designed, had control experiments, and what they, I will discuss this experiment a little bit further, but for the moment, I should say that when that experiment was done, it appeared that the bees didn't have this, they didn't have alleles for the Varroa-specific hygienic behavior, which is remarkable because you find that in all the other populations that have become resistant by natural selection. Then the other observation is that there are very few cases in Europe of resistance to deformed wing virus. And you might expect that after 40 years that the selection would have made bees more resistant against the deformed wing virus. Now let's now look at this experiment of Fries a little bit more closely. The experiment started with 150 colonies and only 13 survived. And Fries had taken care to sample Sweden in a very good way, and so he had colonies that represented Carnica, Ligustica, and buckfast bees. And as I said, the surviving colonies had no alleles for Varroa-specific hygienic behavior, and they had no effective grooming behavior. And that's interesting because a sample of 150 colonies from Sweden is quite a large sample. So that it means that because the 13 survivors didn't have these alleles, that they were absent in the 150 colonies he started with. But because it is a large sample, it must mean that there were very few alleles of Varroa-specific hygiene present in the Swedish population. And that's very strange. Where have they gone? Now, let's look again at these experiments. The surviving colonies were small and they swarmed often. And that are both characters that, of course, help you to be probably that's not easy for Varroa. During swarming, Varroa cannot reproduce, and in a small colony, Varroa cannot reproduce very fast or very long. And the other thing is that Sweden has a very short summer, so the season is already short. They found in the 13 colonies evidence for increased homozygosity and strong inbreeding. Two things were interesting to note that may contribute to a certain resistance. There were fewer reproductive mites in the colonies. Now, one reason why you have non-reproductive mites may be this hygienic behavior. Sorry. Might be that this hygienic behavior does not remove the mites that do not reproduce, but we know that the hygienic behavior was not there, and so we have to look for another factor. And the literature says that sometimes there is something like a brood effect, which may have to do with the pheromones that the bee larvae produce and which are used by a signal if that signal by Varroa, and if Varroa does not receive an enough strong signal, it can wait with reproduction, and so it doesn't breed. This experiment is also one of the 3 published cases where bees were less susceptible to deformed wing virus. Evidently, they were not really resistant, because if these colonies are placed elsewhere in Europe, they die as quickly as non-resistant bees. Now, so we have, with this experiment, we have evidence that in the European population, or at least in the Swedish population, there is a loss of alleles for resistance. Now, my hypothesis is that selection for desired traits, for example, low aggression, high productivity, or low swarm propensity, or whatever you want, has exhausted the genetic variation for resistance in breed honeybees in Europe, and most likely also in North America. But how did that happen then? And when I give this talk to bee breeders, they always tell me that they take good care of the genetic diversity of the bees they produce. And I agree that they do all sorts of things that are good. And we know also that there is, if you want to select bees for a character, there is enough genetic variation to do so. You can always achieve the goals of selection. So it seems on the surface, it seems to all be very well. But why then did those resistance alleles disappear? Now, if you want to know that, you have to think about possible mechanisms by which those alleles can disappear. And the 2 mechanisms I could think of are the loss of rare alleles and selection, and the fact that selection may lead to homozygosity. Now, we are going to review both cases and to see what we can find. But before we do so, we have to understand the mechanisms that bees use in nature, in a natural situation, to cope with pathogens. A lot of that you know already, but we'll go through it, and then you'll discover why we do so later. So first of all, honeybees have only 1 generation per year, and relatively small populations. And bacteria and viruses, they have many generations per day even, and a very large population. For that reason, pathogens have many more mutations per unit time than honeybees, and the pathogens can, by those mutations, evolve new virulent variants. And then bees keep their breeding nest at 35 degrees centigrade, and that's optimal for the reproduction of bacteria and viruses. And in winter, they keep the nest temperature high, which is also good for the viruses and the bacteria. And then thousands of bees live in close contact, and they exchange food, and potentially they exchange also pathogens. So that makes that the whole biology of bees makes them quite vulnerable to pathogens. Now, of course, evolution has served the bees well, and bees are well able to defend themselves against pathogens and parasites. The prevention goes with natural antibiotics. Those are present in propolis, in pollen, and in honey. They are sequestered from plants, and the plants produce these substances for the same reason that the bees use them. And then they can suppress attacking pathogens via behavior, and then we come to this hygienic behavior, the removal of infected larvae or of dead larvae, and the grooming of self and other bees. But there's a third factor, and that is resistance through immune responses. And that's what we will talk all about largely this evening. Now, let's now consider the tools that bees have for defense against infectious disease. And the first one is an extremely high recombination rate. So that is the frequency at which the chromosomes break and exchange parts. We'll talk about that a little bit more very soon. And then the panmictic population structure, which is very important, and other things you know of, the polyandry of queens, and finally the departure of the mother queen with a swarm. Now let's first consider the recombination rate. Honeybees have the highest recombination rate of the entire animal kingdom. That's quite shocking. And biologists always ask, why is that? And we think that it's a product of natural selection. And it's difficult to prove, but what you can do is you can make a big genetic simulation model in which you let evolution go very fast so that you can understand what goes on. And this has been done by Sasaki and Iwasa, and they found that recombination is the answer of slower reproducing organisms to the high mutation rate of the pathogens in the arms race that they have with those organisms. So their conclusion was that only strong selection by pathogens can result in such an extremely high recombination frequency. So what's the function of recombination? It creates new genotypes and it can bring together favorable alleles. More recombination may enhance the probability of this happening, but recombination can also separate favorable alleles. And therefore, there must be an optimal recombination frequency, and it's natural selection that optimizes the recombination rate. Now the panmictic population structure, we had this definition already, so it's a population in which mating can occur between a very large number of individuals, sometimes from a long distance. Do we know if honeybees have a panmictic population structure? Yes, there's very nice research. It's started with the brothers Rutner. They were interested in how drones find drone congregation areas. And what they discovered was that drones from a particular hive, they visit several drone congregation areas. So that already makes that it involves a large area. And then Emmanuel Baudry did a study in which he did, she sampled bees from all the colonies in an area, and she characterized them on basis of DNA samples that she took. And so she could recognize whether bees came from a particular hive. And then she went on a certain day to a drone congregation area. And on that day, there were 12,000 drones there. And she collected drones and she did the molecular biology, and then she knew that the drones came from 240 different bee colonies. And finally, Annette Jensen, she studied this problem also, but she measured the distance that between the queen and drones that mated with that queen. And on the base of that, she could calculate that the drones visiting a congregation area, they come from an area that might be as large as 225 square kilometers. Well, together, these three studies show that honeybees have a panmictic population structure. And why is that important? It is important because in a very large population, you can have rare alleles. And if bee queens can mate with drones that come from a large distance, they can have access to these rare alleles. And these can then be used against novel pathogens. Now, when I tell this story, people always says, yes, but how is this possible? Because bees are also locally adapted to the climate and to the flora. And there's no contrast there because in the study of Annette Jensen, 10% of the matings came from a very large distance, between 7.5 and 15 kilometers. But 90% of the matings were only from less than 7.5 kilometers. So those 10% of matings, they're very important because they can result in the rapid recruitment of new alleles. But the local adaptation is still possible through the majority of matings over shorter distances. So there's no controversy here. Now, the extreme polyandry you're all familiar with. It means that workers in a colony have different fathers, and so it makes that there is a lot of genetic variation among the workers, and of course that can be handy when there is a pathogen going around. It means that not all the bees are to the same level susceptible to that pathogen. Now, then we have the mother queen that leaves the nest, which is remarkable because in the animal kingdom, if you look around, it's always the mother that stays at home and the daughters that disperse. So here again, the honeybee is a remarkable exception. And the question is, why does the mother queen in this case move out? Why doesn't she stay at home and let her daughters depart with swarm? And the interesting thing is that when a young queen takes over the old nest, then the genetic makeup of the colony changes dramatically because she mates with different drones than her mother had done, and she shares only 50% of their mother's genes, and so there's really a dramatic move in the genetics. And the idea is that the alleles, the new alleles that come in, can provide resistance to bacteria that are already present. And there is much more to this story, but I have no time to tell that now. So we have seen that the panmictic population structure and the extremely high recombination rate are both important for the evolution of resistance. What I want you to remember from this evening is that they form a double-edged sword. And that is because you need both to get a result. The panmictic population structure allows the recruitment of rare alleles from a large area, and the high recombination rate makes them that they can rapidly be combined with alleles of other genes due to the high recombination frequency to form new resistant genotypes. And so you really need both. And now we understand the biology of bees that live a natural life, and we can look what goes wrong in the selection of breed honeybees. And of course, it starts with the fact that it isolates the bees from a panmictic mating structure, because often they are mating on particular places with drones of the same breed. And then it causes homozygosity of the genes encoding for behavior. And it causes the loss of rare alleles. Still a hypothesis, but we'll go further with this and I'll explain how that works. First of all, the alleles that you prefer when you select for particular bee behavior, those alleles are often recessive. And so you want them to be you want homozygosity to make that the genes are expressed. Now, let's talk a little bit more about the heritability of complex behavioral traits. Characteristics such as aggression, or high honey production, or swarming behavior, or Varroa-specific hygiene, those are complex behavioral traits. And that means that its inheritance involves many, and then we talk about hundreds of different genes. And many of these alleles, they are recessive, and they are only expressed when an individual is homozygous for it. Thus, as a result, selection for these traits results in homozygosity. And often, if you select on a gene that you need for a particular behavior, the neighbor genes, they hitch a ride and they become also homozygous. So, and in the end, you could have runs of homozygosity in the genome. And why is that not desirable? That is because recombination has no effect on homozygous genes. Because if you exchange the same allele for the same allele, nothing happens. Now, this might be the most important slide of this talk. And it explains the loss of rare alleles. You should know that in a population, the alleles of a particular gene occur at different frequency. Some alleles are common and others are rare. Now, selection is always taking a sample from a large population. And if you take a sample from a large population, then it's far more likely that you select a common allele than that you select a rare allele. And the truth is that almost all aspects of beekeeping involve selection. And often it is repeated in every generation of a bee breed. And that means that with time continuing, you lose more and more rare alleles. Now, for people that are more visually inclined, I show this graph where you can see that on the y-axis there is the probability of an allele being selected, and that probability is low when the allele is rare, and it becomes much higher when the allele is common. I see that I omitted one M from common. Now you might ask, why are there rare alleles and why are other alleles common? Now the common alleles is easy. Common alleles have become common through natural selection. It's natural selection that have made them common. That means that they must be useful under the circumstances. Now, rare alleles currently offer no advantage in natural selection, but they may become advantageous again when the conditions change. For example, when a new pathogen emerges. And we all know that the world is changing the whole time. So in general, it's so that alleles which are rare now, they may be needed in the future because the world is changing. Now, here's some examples then from the way we take samples out of a large population. First of all, breed bees cannot be part of a panmictic population if they mate on a mating station. And nowadays there are people who think that you can select very quickly by using single drone insemination. And it's quick, but also very dirty, because it's the smallest sample you could take. And so it's the most efficient way to lose rare alleles. And then when we collect young work larvae to breed queens, very often we take a number of larvae from the same frame, at least from the same colony. And also that's a sample, and so you lose rare alleles. And then again, very often bee breeders, they use those techniques every generation, and then there's little hope of maintaining the variation for resistance within a breed. So what goes wrong? Beekeepers often work with a number of different selection lines to ensure the genetic variation. But if selection uses a number of selection lines, I mean, even if it would be 20 or 30, to maintain the heterozygosity, the rare alleles will still be lost in each of the lines. So it helps to maintain your heterozygosity, but it doesn't help you to conserve the rare alleles. So heterozygosity is no guarantee for the conservation of rare alleles. We should maybe spend a little bit more time to think about that. I thought to explain this further, we make up an example which is rather extreme, but it serves to make the point. Now, suppose that we have a particular gene in a population that has 35 different alleles in that population. And now we start selection of bees. And we do that for a number of generations. So we sample from this large population, and then finally we have lost 25 scarce alleles. That leaves 10 alleles that are equally common in the selected population. Now, if we now calculate the heterozygosity, then we know that the chance of a fertilized egg being homozygous is only 10%. So even if you have lost more than 71% of the alleles, of the rare alleles, through the selection, the bees are still for 90% heterozygous. So it shows that there are different things. And the other thing which is worth to note is that if you only consider the heterozygosity of your bees, then before that is going to give problems, then you have already for a long time lost those rare alleles. So the loss of rare alleles occurs long before there are problems with heterozygosity. Now we look at a little bit more, because so far I've only talked about breed bees. And now we want to expound a little bit. And that's why I want to talk about the plague of drones. First of all, a healthy hive may produce up to 30,000 drones during a season. And there is a very strange contrast. Keepers of breed bees, they are careful with their young queens. These are kept virgin and sent to a mating station to mate with drones of the same breed. But they release their drones into the wild. They don't need them. And as a result, the native honeybees are threatened by hybridization. But that's not the only bad thing. The thing is that the massive invasion of exotic drones, repeated year after year, also hampers natural selection for resistance to bacteria and viruses of free mating bees. And there are lots of people that do not have breed bees and that have their bees mating in nature. But if there is an enormous number of these breed bee drones that have lost the rare alleles, then that population will also lose the rare alleles. Now, something which is not very much related to what I told you, but it shows another aspect of the costs of selection. And I was shocked to read this paper last year when it came out. Anthony Nierman and Dennis van Engelsdorp, they, for some reason, repeated an experiment that had been done 50 years earlier. And then, by chance, they discovered, because it was not the purpose of the study, that the median lifespan of honeybees in the US was halved, declined from 34 days to 70 days. And they attributed the difference to the fact that Varroa is there and it was not 50 years ago. And of course, there are also negative effects of chemical Varroa control, to which we might add that there is also a problem in the modern agriculture that uses a lot of chemistry, and there a lot of biodiversity has disappeared, so it's more difficult for the bees to collect their food. But it's hand waving. You can say, okay, that might be what they have done. But there's another way to look at this problem. That is that they have been selecting these bees. And they have done it then for a very long time. And because I sometimes work also for animal husbandry genetics, I know that, for instance, cows have been selected for a higher production, and the result is that they have a much faster metabolism. And I did a lot of studies on parasitic wasps, and I was quite surprised to find that these cold-blooded animals, that they differed in metabolic rate, and that this difference was heritable. So I think that if you select for a higher honey production, then you select also for a faster metabolism in honeybees. And a faster metabolism means that you live faster, and so you live shorter. And a faster metabolism would also make it harder to survive the winter, and therefore contribute to winter mortality. Now, this is all a hypothesis, just like Nierman and van Engelsdorp have an hypothesis about why the bees have changed so much. But I think we should study it in more detail, and for now take it as a warning. Because that's what I miss in much of the literature on selection in honeybees, is that one rarely talks about the costs of selection. One tends to focus on the benefits of the selection. Now, there we come to the conclusion already. So honeybees need a panmictic population structure to be able to develop resistance to new pathogens and parasites. Because from this large population, they can recruit rare alleles that may be needed to produce the resistance. And the fact that the evolution of resistance is enormously important in honeybees is also demonstrated by the fact that they have the highest recombination rate in the animal kingdom, showing that natural selection has done a lot to help bees to defend themselves against new pathogens and parasites. And so I think that the loss of rare alleles in selected bee strains hampers the resistance to new pathogens. And there's a problem there because, of course, we need to select bees. Certainly, if you're a professional beekeeper, you want to be able to work efficiently. And I know that beekeeping is an industry in which it's difficult to earn your money. And so, of course, you want super bees that have a high production, are not so aggressive and easy to work with. And so we should always select bees. But at the same time, we should also have a large palmictic population that could act as a reservoir for these rare neutral alleles, because that is sort of an insurance for the future. And nowadays, most beekeepers are amateurs. So for amateurs, a very high production is not so important. So my idea would be that we make high productive bee strains for the professional beekeepers. And at the same time, we try to give the bees their natural way of mating in a large palmictic population, and to make sure that the genetic variation, including the rare alleles, stays large enough. And then anytime the professional beekeepers, for instance, when there is a new pathogen going around, they could go back to this large population and try to find the resistance in that population, and then select new strains of bees. Now, one way to do this is to have our native black bees, and then the large palmictic populations could act as a reservoir for rare and neutral alleles, and we could have those bees in the wild, and we could have those bees with amateur beekeepers, and then there would be no problem that there are beekeepers that have highly productive bees that release their drones, because those drones are of the same subspecies, so there's no problem. And I should add to that for a long time there has been a belief that hybrid bees are superior to black bees, but you should know that the black bee population in Europe is not in a very good shape, but there is still enough genetic variation to breed for any desired characteristics in the black bee. You don't need to cross different subspecies. And then there are many other reasons to preserve our native black bees. And with that, I think I call it a day. Okay.
All right. Thank you very much, Jack. You've got to that's it. Yep, we're back now. Well, at the moment we've got one question come in. But may I start with something, please? Because I've had a look at the attendance list, and there are several beekeepers who keep their bees with small closed populations. Now go back to the work by Ingemar Fries. You suggested there was strong inbreeding there, but there were 150 colonies of 3 different types, which I assume were brought in from outside. If that's causing inbreeding, let's say, or there's inbreeding there are quite a lot of small isolated populations, which could be islands, they could be peninsulas, or they could even be where it's sort of marginal for bees to be kept. Therefore, the population level is very low. Are they in any particular danger, do you think?
I don't think so. The literature on in conservation biology says that well, if they're really isolated on a small island and there's no influx at all, then there might be a problem. They may become highly inbred and lose viability. But if there is the equivalent of one new queen or one drone from outside coming in each year, then that is enough to counteract the loss of heterozygosity in this case. So you don't need an awful lot of immigrants.
No, but in the Swedish experiment, they had 150 colonies.
Yeah, but then
which...
only 13 survived, and that's where the problem started. Then
yes, I'm there with you. Sorry, I thought the inbreeding was 150 colonies. Sorry, apologies about that. Right, now we've got one question. At what rate do honeybees' chromosomes mutate?
yeah, I don't know. I have no idea. But if you look at the difference in number and the difference in generation time, you talk already of differences between bacteria and bees that they are in an order of more than 1 million. So if the actual mutation rate would be low, or even if it would be high, it would not help the bees.
Okay, so the second part of the question then is probably not needed, which was, is this another way of generating those rare alleles? So, presumably
Oh,
that's,
yeah,
yeah,
of
carry
course,
on.
all the alleles have come into existence by mutation. But you may be talking on a time scale that is much longer than we find interesting as beekeepers. It may go over maybe the millions of years or thousands of years that these alleles have been created. But once they're there, we don't want to lose them. I mean, there's three types of alleles. Of course, if there's a mutation, it may be a harmful mutation, and then it's selected away, it disappears. But then you have neutral alleles, and they're not harmful, but they could become useful when the world changes. So what you want is, as a sort of an insurance against changes, you want to have these neutral alleles floating around in the population. And if you have a small population, then by chance, not only by breeding, but also by chance, you lose a lot of those alleles. There is a lot of literature in the conservation biology on the loss of rare alleles when populations become too small. And it's similar, but in fact, what bee breeders do is they mimic small populations. And so that's why I decided that we have to be careful with that.
Okay, the next one is, what advice will Jacques give the beekeepers tonight to encourage the distribution of these rare alleles in their colonies and apiaries?
Yeah, but we don't know those rare alleles. So the only way we can encourage their existence is by taking care that at least a major part of the bee population is mating in the blue sky and has a pannictic population structure. That's the only way you can do it. I should add to that some people sometimes ask me to say, well, yes, but if you take a different subspecies of bees and you hybridize those, then you get lots of new alleles. Which is true. I mean, that has been measured, and it's certainly true. The only problem is that a lot of those alleles that you then get extra, they are not functional in the sense that they might represent advantages in countries with a very different flora or a very different climate. And so they would go lost anyway. So I'm in favor of keeping the native bees that have probably most of those rare alleles which are useful in our climate, and not hybridized bees.
Okay, another one. Somebody has, I have more info on the Gotland colonies. The figures of 13 survivors is very old, 4 to 6 years after the experiment began. Recently, I tried to track down how many there are now. Barbara Locke is being cagey and says a new paper is out soon. So I suspect the population has rebounded. So it's really just a sort of, yeah, a comment. But would you like to say anything on that? Do you know any more about the
I know that these Gotland bees have been their breeding has continued, but of course that you cannot hybridize them with other bees, because then you don't have the Gotland bees anymore. So they remain very much inbred, and they are sometimes used in experiments elsewhere in Europe. They have been brought to the Netherlands, for instance, for an experiment on natural selection. But that's not very useful, I would say. I think the good thing is that they have kept those colonies, is because that gave them the time to do all that additional research on the genomics, and so we now know far more than Fries has ever known about his bees.
There's one here which is applicable to the UK, but I think we can take it as Europe wide. Do we know if the shortened lifespan found in the US bee population is also found in the UK? For that, read Europe, Jacques, I think.
Yeah.
Maybe there have been no such studies as yet.
Yeah, I've been looking. I was very curious. Of course, it's the first thing you ask yourself. Are our bees also much shorter lived? And I couldn't find any published data on that. Of course, it was a rare chance that somebody repeated an experiment exactly in the way it was done 50 years ago.
Yeah.
And I would think that because the beekeepers in America, they're far more professional, and many of those also because they have now the Africanized bees in the southern states. They replace the queens every season again, and so I think that those effects would be probably stronger in the US.
Okay, is treating our bee colonies with chemicals to remove varroa, reduce of varroa, I think is probably more accurate, actually harming the bee's ability to naturally select beneficial alleles?
Yeah, the short answer is yes. The problem is that natural selection is selective mortality. And so as soon as you don't like that your bees die, then you start to treat against varroa.
Yeah.
And of course you hamper natural selection. Good natural selection is you let your bees die and you see who survives.
Should we avoid selecting for positive traits such as temper or honey production and instead aim to maintain diversity?
Yeah, but what I said at the end of the talk was that you should not do that. But I think that because professional beekeepers, they need those bees. So we should do that. But
Yeah.
if everybody would keep that type of bees, then the bees would go extinct. So I would say let the majority of people not use selected bees and let only the professional beekeepers do that.
Yeah. In fairness, Jacques, this question might have been written before the slide came
Yeah.
up. So,
Yeah, no problem.
There, okay, coming in fast now. There is don't don't let me stop you. Keep bringing them in. There is a suggestion that when bees choose an emergency larva to raise a queen, so obviously emergency queen, they choose ones from rare patrolines. Could this be a defensive measure to retain diversity?
Yeah, it could. But unfortunately, we know very little about those resistant genes and those rare alleles. If we would, I think in the years to come, we will know more about the molecular genetics of bees, and then we could positively answer that question. We cannot now, but I believe, but my belief, I'm really a believer in natural selection. So that means that every time I see something done by bees, I ask myself, what is it for? What's the use of it for the bees? And in this case, also, if new queens are selected from particular patrilines, then it's possible that that serves somebody. Maybe only the workers who had that father. We don't know. Maybe the whole colony. We don't know.
Right, this is one you could probably write a book on. How do we improve mating with black bees with so many hybrids being around?
Yeah, I'm trying that with some colleagues in the Netherlands, where there are very few black bees left. And what I find when I'm going around and I speak to beekeeper clubs is that there are lots of beekeepers now who are convinced that it's better to have your bees mating in nature And so most of the people, they have hybrid bees, and they would like to have black bees. And I tell them that it's not possible right now because there are no black bees, not enough to serve everybody. So that the only way to do it is a gradual change. We should improve increase the number of black bee colonies. We should improve the genetic variation of those black bees. And then slowly we hope that we can saturate the bee world with these black drones, and in that way get our black bees back.
Well, you would also populate the free living population as well, wouldn't you?
Yeah, that's another initiative in the Netherlands. We have very few good old forests with hollow trees, and so there are several people now who are placing nest boxes, which are formed from hollow tree trunks in forests to make that we get bees in the wild.
I was interested to hear that some queens are now inseminated with semen from only one drone. Is it therefore beholden to beekeepers buying in queens from commercial breeders to ask more questions about the breeding program involved, especially as their drones will affect local populations?
Yeah, it's very difficult to, if you buy a queen, it doesn't say in the small print with whom she has mated. It should
No,
be.
they don't give you much information at all, do they?
Yeah. But some standards protocol would be nice, yes.
Yeah. Okay, well that's all the questions that have come in now, Jacques, and we've gone over the hour, so just remains for me on behalf of the viewers to thank you very much for a very interesting talk, and we'll look forward to seeing you at the National Honey Show. Thank you very much indeed.
Well, thank you for listening and for the discussion. I really enjoyed it, and I hope to see you all in October.
Thank you very much, Jacques.
Okay,
And
bye-bye.
good night, everybody.
Good night.